Hello, good afternoon, everyone. Let's get started. This week, we're going to be doing the blood and lymphatic system. You do have a quiz next week, and that's only on lecture number 1, which we'll get into more details afterwards when more people show up. I'll tell you, how many questions to expect and so on and so forth. But let's get started with lecture 2 with a lot of things to cover in this study guide. Okay, first question. List 3 types of granulocytes. Okay, so just to refresh your memory of what a granulocytes are. Okay, so granulocytes, they are basically a type of white cells that has visible granules, okay, under the microscope. Okay, and granules are basically kind of related to lysosome. You might remember lysosome from last semester. They are these little vesicles that has digestive enzymes in them. So granules are kind of like that. They could have digestive enzymes. They can have oxidizing agents in it. And you will be able to see them clearly when you stain the granulocytes, okay, and you look at them under the microscope. So there are 3 types of granulocytes in general, and I see some people are already writing down what they are. So the first one is the neutrophil, okay, and Adriana mentioned eosinophil. That's another one. Eosinophil. And what's the last one? Basophil. Thank you. Okay. Now, just to give you a little bit of refresher of what each one of them does, or kind of like a characteristics of them. So the neutrophils are the most abundant granulocytes. Okay, roughly 70% of your white cells are neutrophils, and they are also considered as a type of phagocytes. Okay, so they go around and they basically eat things up that are not supposed to be there. Eosinophil, on the other hand, they are responsible for secreting chemicals to fight off parasitic infection. Okay, fight parasite. Now, they also do other things like, they're responsible for allergic reactions and so on and so forth. But, I'm just trying to keep it down to one thing for each to help you remember. All right. So basophil, what is basophil for? Well, they are responsible for the inflammatory response. Inflammatory response. Okay. And, just in case you're wondering, you will learn this next week as well. But just in case you are interested right now, the characteristics of inflammation. Okay. So that includes things like redness. Like if you cut your finger, it's going to be red. It's going to be painful. It's going to be swelling. And it's going to feel a little bit warm. Okay. So these are the hallmarks or characteristics of inflammation. And part of it is you can blame it on basophil. Okay. Basophil. So there you have it. That's the 3 types of granulocytes. Okay. What about agranulocytes? Okay. What's the difference between granulocytes and agranulocytes? Well, these agranulocytes, they also have granules. Okay. They also have granules, but they don't really soak up the stain when you stain the slide, and so you can't really see them. Okay. But are not visible under microscope. Okay. So it's a morphological distinction between granulocytes and agranulocytes. Like, can you see them or can you not see them? Three types of agranulocytes. Anybody want to help me out here? Three types of agranulocytes. Yeah, thank you, thank you. We have the monocytes, which are kind of, the inactive cells. They just patrol around the body, checking here and there, and then, want to see if there's anything wrong. And if they detect something is wrong, they will transform and change to macrophage. Okay. It's a one-way transformation. Okay. Sometimes you might have organs that are very exposed, very vulnerable. So in those places, they are going to stay as macrophage. Okay, and you kind of skip the monocyte stage. They directly transform into macrophage and just stay there. We call those resident macrophage. That means they live there, basically. They stay there all the time. Another example of agranulocytes would be the T cells and the B cells, right? Okay, so T cells, B cells are examples of lymphocytes. They are made in the lymphatic system, which we'll talk about later. And T cells will coordinate the immune response, while the B cells, they will make antibodies. So let's just match these up. Okay. Responsible for inflammatory response. If you have been paying attention, you will know that it is the basophil that does that. Coordinate immune response, that is the T cell, can differentiate into phagocytic macrophage. Differentiate is just a very, very fancy word for transforming. Okay, and these are one-way transformation. Okay, once you become a macrophage, you stay that way. And that will be monocyte. Attack parasites. Okay, we mentioned it's the job of eosinophil. Most abundant phagocytes. You have a lot of them, very common. That is neutrophil. And the B cells are the ones that produce antibodies. Okay, so antibodies are these Y-shaped proteins that would tag and recognize antigens that are on the surface. Okay, so like if you have a virus here, okay, and then you have all these proteins. Okay, like if you think about the COVID virus, the SARS-CoV virus that causes COVID-19, right? They have all these spike proteins on top of it. So you will make antibodies that would recognize them. There's a key and lock relationship between the antibodies and their target. Okay, and the cell that is responsible for making these antibodies, that is the B cell. Is everybody okay so far? Any questions of what we are talking about what we are discussing here? All right, moving on. List the steps in the coagulation pathway, starting at release of thromboplastin from injured blood vessel. Yeah, so typically speaking, we have our blood vessels, and last week I told you blood cells like a smooth surface, right? So they would just be floating by, no problem. But if you damage this blood vessel, okay, by inserting my favorite type of injury, a thumbtack in there, then you expose all the underlying tissue, right? All the collagens and stuff like that. And those damaged tissue will release something called thromboplastin. Okay, so normally you would not have thromboplastin. It's only there if you damage your blood vessels. Okay, so in the presence of thromboplastin, along with a little bit of calcium, what we will do is we will convert prothrombin, okay, which is inactive, into the active form, thrombin. Okay, and then it's going to be a cascade of events, meaning it's like domino effects, right? One thing leads to the other. The thrombin, along with, again, some calcium, is going to convert the fibrinogen into fibrin. Okay, so just to reiterate, this is inactive. This is active. Okay, so do you have thrombin in your system right now? Not unless you have some kind of cut. Okay, in your bloodstream, you should only have the inactive prothrombin. Same thing with the fibrinogen. This is inactive. And in the presence of thrombin, it will become active. Okay. Now, this is a simplification of the coagulation pathways. There are other coagulation pathways that occurs within the body. Just so you know, in case you read about it and you're like, oh, is all this a lie? Well, it's not. It's just there are multiple things happening, and I choose the easiest one for you to learn. Okay. So over here, once you form the fibrin, like what exactly are they, right? The fibrins, they are basically, little strands of proteins. Okay, and the strands will come together and form a mesh. So this mesh will trap red cells. It will trap platelets. Okay, and then eventually it will seal the plaque, assuming you have removed the thumbtack, of course, and it will begin the healing process. Okay, so this thing here, that's the fibrin clot. Okay, and then eventually it will heal, unless you start picking on the scab, right, and then it bleeds all over again, and you will have to wait for it to clot one more time. All right, so make sure you know the order of this process. Okay, complete the following chart related to ABO and RH blood group. Let me just quickly refresh your memory about what blood groups are. Blood groups. Or does anybody want to tell me, based on their understanding, what exactly are blood groups?
Blood groups are presence or absence of antigens on red blood cells and antibodies and blood plasma.
Yeah, basically it, right? Thank you. That is exactly in fact, it's an exact definition I give in the PowerPoint. So these are the presence or absence, right, of antigens on the surface of red blood cell. Okay, so antigens could be proteins, could be carbohydrates, could be glycoprotein or glycolipids, right? Sugary proteins, sugary lipids. So whether you have certain types of proteins or sugars on your cell or not, on your red cells or not, that's going to determine your blood groups. And there are many blood groups that exist in the human population, right? At least 30 blood groups exist. We are only concerning ourselves with 2 of those blood groups, the ABO as well as the RH. So how many alleles do we have for the ABO blood group? Does anybody remember the number from the lecture? That's right, we have three alleles that determines the ABO blood group. We have the IA allele, the IB allele, and the little I allele. So a total of 3 separate alleles. And we can combine them in various ways to form different genotypes. Remember, we are diploid organisms. So even though there are 3 alleles that exist in the population, you can only have a maximum of 2 alleles in your genome, right? So I can have IA, IA small I, IB, or IB small I. I can also have IAIB or little I. So a total of 6 possible genotypes. Genotypes are genetic makeup of an individual. So of the 6 genotypes, how many phenotypes would there be? Phenotypes is the expression of your genotype, right? That's you know, what kind of blood type you actually end up having in this case. So there are 6 possible genetic combinations. How many different blood types can you have in the ABO blood group? Yes, there are 3 types. So you could be type A, you could be type B, you could be type O, and there's actually one more. Yeah, AB is the other one. Okay, so a total of 4 different blood groups. Okay. So how do you get that? Well, this is because IA and IB are dominant. Remember the dominance and the recessive stuff that we learned last semester? Right? IA and IB are dominant over little I, and IA, IB are co-dominant with each other, right? So when you get an IA and an IB together, that's going to give you type AB blood. If you have IA combined with a little I, you will be type A blood. And so these two will give you type A blood. These two will give you type B blood, type AB, and then little I is just type O blood. Type O blood is the absence of A and B on the surface of your red cells. Okay. Any question about this stuff? If you're confused, please ask. It's only going to get more confusing because we need to build upon this knowledge. Okay. Can you review once again? One more time, okay. Which part? The whole thing or just the last part? Just the genotype? Okay. So we have 3 possible alleles, right? These are the 3 alleles that are related to the ABO blood group. Okay. So we have 2 chromosomes, right? We have 2 chromosomes for each type. So 2 chromosome 1, chromosome 2, so on and so forth. So on the chromosome where you will find this particular allele, you would have 2 slots. Okay, so these 2 slots, what could you put in there? Well, you could put an IA, you could put an IB, or you could put a little I, right? So if you try all the different combinations, they would be all the ones that are listed here. Okay, you can get 2 of the IA, you can get an A with an I, 2 Bs, a B with I, and so on and so forth, right? So that's your genetic makeup. Okay. So what does it mean to have the IA allele? Well, remember these alleles are genes, and genes contains the instruction to make the protein, right? So if you have the IA allele, you actually have the instruction to make the A protein, we'll say, okay? The A protein, you can make that and you'll put it on the surface of your red cell. So if both copies are just the IA allele, then you can only make A proteins, right? So as such, you will have type A blood. If you have IA and small I, you have the instruction to make the A protein. I does nothing. Okay, I has no instruction. It doesn't have instruction to make anything. So having this combination will also give you type A blood. Same situation for B. If you have both copies of B, then you would there should not be a comma there. Then you would be type B blood, right? BI also type B blood. Right? 1 copy that tells you how to make the B protein. And again, the I doesn't do anything. IAIB means you can make both A and B. That makes your blood type AB. And then little I is nothing. Think of them as cookie cutters. The A is to make star cookies, and the I is to make moon cookies, and I is no cookie cutters. So if I give you, I forgot what I say. I said star and moon. So if I give you 2 star cookie cutters, you will only be making star cookies. Right? If I give you 1 star cookie cutters, you will also only be making star cookies, which is why both of these will be type A or star cookies. Right? If I give you both the star and the moon cutter, then you can make AB, right? You can make both types. Does that make sense? Any more follow-up questions? Okay, great. And Alex, you okay? All right, next part. We're moving on to the RH. Okay, so RH is an other blood group. Remember, we have 30 different blood groups, right? So RH, there are only 2 alleles here. Okay, we have the positive allele and we have the negative allele, a total of 2 alleles. So how many different combinations can you have? Well, you can have positive, positive, you can have positive, negative, or you can have negative, negative. Okay, 2 alleles give you 3 possible combinations. What about your phenotypes? Well, do you have the Rh protein or do you not have the Rh protein? To make the Rh protein, you will need the instruction to do that, right? And that's what the positive allele does for you. It tells you how to make the Rh protein. So if you have both Rh positive copies, then you will be Rh positive. Even if you just have 1, that's enough, right? Think of the cookie cutters I'm telling you, right? Like giving you 2 moon-shaped cutters versus 1, it doesn't really matter. If you can make the moon cookies, then you can make it, right? It's about whether you can make it or not about how much you can make, right? So here you can make the Rh proteins and put it on the surface. That will cause you to become Rh positive. And if you are negative for both, then you will turn out to be Rh negative. So 2 different phenotypes. Okay. Moving on. Now this part I already did in class in the lecture recording, but you know what, let's just do it again just to help everybody, you know, review or bring you up to speed, or you know, if you haven't seen it before, haven't watched it yet, it's probably going to be beneficial for you to look at this. So again, we're just looking at the ABO system right now, not even looking at RH at the moment. And we want to know what kind of genotype makes up a particular blood type, what kind of antigens are on the surface, and what kind of antibodies are in the plasma. And in case you are confused with all these words, remember, if you take blood and you spin it down, okay, if you spin it down, you will get the hematocrit down here. This is where your RBCs are going to be, okay? And then on top, you're going to have this liquid portion of the blood. And the liquid portion of the blood is what we call the plasma. Okay, so antibodies, they are found in the plasma. Okay, these are antibodies. All right. What's the difference between antigen and antibodies? Does anybody want to clarify that for me and for the class maybe? Okay. What is the difference between antigens and antibodies? Are they different?
Yeah, the only difference I can make is that the antibody is found in the plasma and the antigen is found in the red blood cell. That
is correct. Okay, that is correct. The antibodies, you find them in the plasma, and the antigens, you will find them on RBC. We can find antigens on any cells, actually. But for the purpose of blood group, we are talking about the antigen specifically that is on the RBC. Okay, so in general, this is true. Your immune system will make antibodies to attack any foreign antigens. Okay, that's an important thing to know. Okay, so if you have an antigen in your system that doesn't belong there, okay? Let's say you take someone's kidney and then it was transplanted into you, then it's full of foreign antigens, right? Then your body will make antibodies to attack that, okay? If you get a virus, you get a bacteria in your system, they are full of foreign antigens, then your immune system will make antibodies to attack them. Okay, that's the key thing to understand here. Okay, so let's take a look at blood type A. What are the genotypes for type A blood? Well, there are two ways to get type A blood. I told you it can be IAIA or IA little I, right? So what kind of antigens will you have on the surface of the red cell? Okay, so if I draw the red cell, which looks like a happy face without the eyes, then you would have the A protein on the surface. What kind of antibodies will you have in the plasma? Remember, you will make antibodies to attack anything that you do not have. Okay, so if you type A blood, you do not have the B protein, and so you would have anti-B antibodies. Okay. Now, there is no such thing as anti-O because O is nothing, right? I told you O is the absence of A and B. So there is no such thing as anti-O. You cannot make an antibody that attacks nothing, right? Does that make sense? Okay, we'll talk about who you can donate to in a second. Type B is IBIB or IB little I. And then on the surface of the red cells, you would have B antigens. And what kind of antibodies will you make if you are someone with type B blood? Yeah, you will make anti-A antibody, right? Anti-A. Okay. Type O. The genotype for type O is little I, little I. The absence of A and B. So there's nothing there. And you will make antibodies for both A and B. Anti-A and anti-B, right? And for AB, it's IAIB. That means you will have both A and B. What kind of antibodies will you have in the plasma in this case? None is correct, right? You don't want to make anti-A because you will end up attacking yourself. You don't want to make anti-B because you will be attacking yourself as well. So no antibodies related to ABO blood group. Okay, when I say none, I don't mean like zero antibody at all, right? Like if you get chickenpox, you will still have like anti-chickenpox antibodies, right? I'm just talking about in the context of the ABO blood group. Okay, so let's talk about blood donation briefly. Regarding blood transfusion, okay, RBC transfusion. Now, I mentioned in the lecture, right? You do not transfuse the whole blood. You can, you know how in movies you see one person just hooked up a line directly from their veins to another person and, you know, transfuse the blood that way. That is not going to work in real life, okay? That's probably going to very likely harm the person who's receiving the blood. In real blood transfusion, we have to separate the RBC from the plasma, and you actually have to filter out the white cells and all that. So now we're just focusing on the RBC transfusion, okay, just looking at the blood cells. And so you have to answer 3 questions. You have to ask yourself 3 questions to determine whether it's safe or not. The first question is, what are the antigens on the donors RBC? Okay. Second question would be, what kind of antibodies are in the recipient, the person who's receiving blood? The recipient's plasma. And number 3 is, will the antibody attack the RBC? Okay. Those are the 3 questions you must ask yourself when you try to figure out whether it's safe or not. So for example, is it safe to take AB blood and give it to a B person? Okay, let's just do a quick poll, right? Gut feeling kind of thing. Okay, what do you think? Is this safe or not? If you think it's safe, please choose A, safe. B, don't do it, not safe. Okay, what do you think? What's your gut feeling tell you? Okay, yeah, most of you got it. Yeah. Very good. Almost all of you said it is not safe. Okay, there's some people who say safe, that's okay. But most of you said it is not safe. Why is it not safe? Well, let's try to answer these questions. The antigens on the donor's RBC. So here is the AB. Right. So what kind of antigens we have on the surface? Well, we have A and B. Right. That's what AB means. For someone who is type B, what kind of antibodies do they have? Well, you can look at the chart and see that, type B people make anti-A. And if you don't want to look at the chart, you can think about it. People who are type B will need antibody against things that they do not have. So they do not have A, that means it's going to be anti-A antibodies, right? So the letter on the antibody will tell you what they recognize and attack. So this guy has an A on it, right? So these antibodies will attack and it will destroy the cells. Therefore, it is not safe. Okay. So generally speaking, that's how you would, use logic to answer the question. But, if you're looking for a quick reference to do this, on a test or something, you can use this, diagram. You put it on a sticky text, stick it on your monitor during the test or something. It will help you. So this tells you who you can donate to. Of course, you can donate to your own group. Right? If you're O, you can donate to all the other O's. If you're A, same thing, AB to other ABs. And it will also tell you who you can donate to. So if you type O, you can give it to A, you can give it to AB, and then you can give it to B as well, right? Okay. A can only give to AB. B can only give to AB other than themselves, of course. Okay, and AB can only give to other ABs. So over here, if you're type A, you can donate to other A's or AB. If you're B, you can donate to other B's or other ABs. O, you can donate to everyone. And AB can only donate to AB. Any questions? Okay, you sure, guys? Let me see if I have any. Okay, let's do some poll questions first before we go on any further. These are pretty straightforward ones, but you know what? Let's do them anyways, since I have them here. Go ahead. Let's do the first one. I'll give you 30 seconds. Okay, so inflammatory response, all you have to do is look at what we wrote down earlier. The correct answer is going to be basophil. basophil. Okay, here's the next question. You got to read the question carefully. It's not asking you what fibrinogen becomes. It's asking you what activates fibrinogen, right? So if you look at the chart here, to activate fibrinogen, we're going to need thrombin. Okay, so the answer is E, thrombin. All right, this next question is tough. I will give you some time to think about it. Okay, go ahead. Okay, just maybe half more a minute for anyone who's still thinking. All right, so we have answers from all the choices. Let's take a look. There are two alleles, LM and LN. If the heterozygous condition is codominant, how many different phenotypic expression will there be in the MN blood group? So let's write out the genotype first. Can someone tell me what the genotypes are? If you have LM and LN as your as the only two alleles, what are the genotypes? I mean, you can't really do the question without writing down the genotypes, right? Anyone at all? Awkward silences, but that's okay. I'm gonna wait a little bit, okay? Okay. Yeah, Tyler got some answers there. Okay, so for genotype, there is the LM, that's one possibility. LM, LN, that's another one. And LN, LN is the other one. There is no LN, LM because it's essentially the same thing, right? Okay. So these are the three possible genotypes. Okay. So if you are LM, LM, what kind of blood type will you be? Right. So that's the phenotype. That's what we're trying to figure out in the question. So if you're LMLM, that means you're a type M blood, right? If you're LMLN, because it is codominance, it means both of them show up. So this would be MN blood, kind of like the AB situation we have in the ABO blood group. And then LNLN would just be type N blood. So there are 3 possible phenotypes. Okay, I'll take any questions. Some of you must have questions for this, right? Okay, what if I say, what if LM is dominant over LN? Then what would the answer be? Let's try one more time. Oops. I don't know what I just did. Oh, I downloaded the results. Okay, here. Try again. Okay. What if LM is dominant over LN? Then how many genotypes, phenotypes are we going to have? All right. Of the 8 people who answered, we got it right? Because if it's dominant, then M is still M, N is still N. But when they're together, the dominant one is the one that's going to be show up, right? So 2 possible genotypes. Okay, so expect some of these questions on the test. Okay, let's continue with this thing. Now, you might have heard people say type O, people with type O blood, they're universal donors because you can give it to everybody, right? But that's actually not true, okay? If you're type O, you can only give it to everybody within the ABO blood group, but with blood transfusion, we have to consider all the other blood groups, right? So, you know, in fact, type O people you cannot literally just take a type O blood and transfuse it to everybody, okay? Because you have to consider other things. So we will have to take a look here at what happens when you consider someone with the positive and negative blood group as well. Sorry, can you explain, please? Oh, like this one, the poll question? Okay, which part? The second part or the first part? Okay. You understand what dominance mean? Right? Remember, like, you know, big B is brown eyes, small b is blue eyes. So like, you know, big B is it's brown eyes, and then small b is blue eyes, right? What about big B, small b? What would the eye color be for someone who's big B, small b? That should be something that we learned last semester, right? Yeah, this would be brown, right? And why is it brown? Because big B is dominant over small b, right? It will cover it up, right? So here I'm asking, what happens if LM is going to cover up, right? It's dominant over LN, right? So nothing changes for the first one and the last one. That's like the big B, small b, small b. That's the easy part to understand. But when they're together, right? When LM and LN are together, since LM is dominant over LN, right? Only the M shows up and not the N. So there are only 2 phenotypes here. Does that answer your question, Mihiret? Okay, great. All right. So in general, you can use this transfusion chart for the ABO blood group. I'm just going to copy that and put it right here. And then I'm going to put negative to positive. Okay. If you're RH negative, you can give it to someone who's positive. but not the other way around. Okay, so let's think about who A positive can receive RBC from. Okay, so if you're A positive, let's focus on the ABO blood group first. If you're A, you can receive from other A's, and you can also receive from O. And if you're positive, you can receive from other positives as well as other negatives. Okay, so if you're A positive, these are all the blood types that you can receive blood from. Who can you donate to? If you're A positive, you can give it to other A's, you can give it to other ABs. But positive can only give to other positives. Okay. Similarly for B, I'm just going to write them down. It's the exact same explanation. What about AB positive? Who can you receive from if you're AB positive? Well, if you're AB, you can receive from all the people in the ABO blood group. So from A, from B, from O, from AB. And if you're positive, you can receive from positive people as well as negative people. Okay, so if you're AB positive, when it comes to red cell transfusion, you've hit the jackpot. You have a much larger pool for selection compared to the others. And who can you give it to? AB positive can only give to other AB positive. O positive can receive from O positive or O negative, and they can donate to A positive, B positive, AB positive, and O positive. Okay. A negative can receive from A negative and O negative. But if you're negative, you cannot receive from positive people, right? Okay. But you would be able to donate to both positive and negative. Same story for B negative. If you're AB negative, you can receive from, okay, you can see this chart, AB can receive from all the blood groups. Okay, but if you're negative, you can only receive from negative. And you will be able to donate to AB positive and AB negative. And last but not least, if you're O negative, you can only receive from O negative. And then you can donate to a bunch of people. Right? So you see, are type O blood universal? No, right? If someone is A positive, is O positive rather, they cannot donate to like AB negative, for example. Okay, so O is only universal within the ABO blood group. Once you consider another blood group, then it's no longer the case. Okay, then you might ask, is O negative universal? Well, not necessarily. What if I also consider the MN blood group, right? What if I consider the Duffy blood group, right? The Cal blood group. There's so many blood groups we have to consider, and every time we do blood transfusion, you know, someone has to match the blood. Okay? That's the job of the technologists in the lab, to you know, match the blood. Something that happens behind the scene. Any question about this huge chart? Okay, let's do random. But do you think the blood type grouping would change in the near future? Probably not. I mean, these are pretty ingrained in us, right? It's not something that would change. You know, if there's any mutation to these things, it's probably you know, gonna get selected against. So I don't think so. Good question though. Okay. A person with A negative blood can receive RBC transfusion from, select all the correct answers. So try not to look at the table we just did. See if you can pick out the correct ones. Okay, let's see. If you're A negative, remember, negative cannot receive from positive people. So automatically that's not the answer and that's not the answer. Okay, so you can receive from other people of the exact same blood group. A cannot receive from AB because they have the anti-B antibodies in them and they would attack the incoming AB cells. That's not the answer. And they can also receive from O negative. Okay, so the answer is A and D. That's what most people choose. Fantastic. Okay. Here's another chart. I'll give you a moment to try that out. Okay. We have the donors here. We have the recipients here. So can A positive give to A positive? Of course, it's the same blood type, so it's safe. What about A positive to AB negative? Okay, so you just go through the table and we will take it up in a minute or so, minute or two. Okay, go ahead. If you're wondering what are we doing, I'm waiting for you to fill in the chart yourself, okay, before taking it up. Okay, so these ones are all the ones that are not safe, and these are all the ones that are safe. Does anyone want me to explain any particular one? Please let me know. Like, why is it safe for all of them, Adrian? Okay, so O negative, right? O negative. What is on the surface of O negative? There's no A, there's no B, and there's no RH, right? So if there is nothing on it, when you transfuse it to all these people, it doesn't matter what kind of antibodies they have, it wouldn't react because there's no surface antigen to react against. That's why it is going to be compatible for all of them. That's the explanation. But in terms of like doing the questions, you can look at the chart, right? You can see O can be given to all the other blood types and negative can be given to positive, right? So that's why O negative can be safely transfused into all these people. Does that answer your question? Anyone else have other requests? Any follow-up questions? Okay, let's do another poll question. Okay. Oh, wrong poll, sorry. Which of the following is a possible blood type? Yeah, okay. Go ahead. You should not be doing Punnett square. That's not how you do this question, okay? Okay, anyone else? Just gonna be closing it soon. Okay. Here are the results. So how do you do something like this? Well, there are a couple of things to remember. I think I wrote this down in the lecture, but clearly either people don't remember it or they need a refresher. So there are 4 things to remember. 4 things to remember. In addition to a million other things you have to remember, there are 4 things to remember when it comes to these kind of questions. Okay, number 1. An individual, a person, would you say a person? A person with type O blood cannot have a child with what kind of blood will they not be able to have? Regardless of who they marry, okay? What kind of blood? That's right. Okay, a person with type O blood will not have a child with type AB blood. That's not going to happen ever. Doesn't matter who you marry. Because if you are type O blood, you are little I, little I. In order to have an AB kid, right, the kid would have IA and IB. That means you need to contribute either an IA or IB. But if you are type O blood, you do not have those alleles. Therefore, you will not have a child with AB blood. That is important to remember and understand. Second thing to remember, a person with type AB blood cannot have a child with what kind of blood? That just means same words. I'm being lazy. I don't want to write it out again. So if you have AB blood, what kind of kid will you not be able to have? You will not be able to have type O kids. Because type O is little I, little I. You do not have a little I to pass on if you're AB. Okay? Seriously, any questions with these 2 statements? Before I tell you 2 more? Okay, very good. Number 3, 2 type O individuals can only have type O children. Okay, if they are both O's and there's only little i's in the family, and the whole family will be little i's, right? The whole family will be type O. And lastly, 2 Rh negative individual can only have Rh negative kids. Okay, same reason. If you're Rh negative, that means you're negative, right? As a genotype. And you know, if you marry someone else who's also negative, then the whole family is going to be negative. If you remember these 4 things, then you can do these type of questions very quickly. Okay, we have an O negative, and then we have an AB negative person. So automatically, I know this is not the answer. I know this is not the answer because type O people cannot have AB kids. Okay. What about AB? If you're AB, you are not going to be able to have type O kids. So immediately I eliminate 3 possible answers. Sorry, I'm just going to try to fix this. I'm going to pet peeve with these things. There we go. Okay. Oh, did I just mess up the whole thing? I totally did. Never mind, we'll not fix it for now. Okay. What about, is it C or is it D? E. Well, they are both negative, and 2 negative individuals can only have negative children. So that's why the answer is just C. Does that make sense? Do you guys understand how to do something like this in the future? Okay. And you don't want to waste time drawing Punnett square and stuff like that, okay? Yeah, I know it's a lot to take in. Alrighty. Let's move on. Did you guys copy this thing? Take a picture here, okay? Take a picture of these 4 things before it's gone. Okay, if you need to. Okay. Next question. Josh's father has type O blood. If Josh has type A blood and he marries Kat with type AB blood, find the expected blood type of their children. Use the Punnett square provided for your solution. So let's do a Punnett square and a pedigree. Okay, here's Josh. Okay, Josh is type A, and then he's gonna marry Kat. Who? What's going on with Kat? Kat is AB blood. Okay, and you know, we know something about Josh's father, who is type O. Okay, so why do we have to involve his father? Who cares? Why do we need to know his blood type? Well, we care because for Josh, who is type A blood, right? We know he has IA as one of the alleles, but is he IA, or is he IA small I, right? That's why we needed to know his father, right? Tyler said Josh is IA little I. That's correct. Because if his father is little I, to get the type O blood, right? Josh is guaranteed to inherit one of the small I, right? So that's how we know he is heterozygous and not homozygous. That's how we know he's IA small I and not IA. Okay, for Kat, it's a lot simpler. He is she is type AB, so she's just going to be IAIB like that. So now we're going to do a Punnett square. For Josh, half of his sperm will carry the IA allele, the other half carries the small I. For Kat, half of it carries IA, the other half carries IB. So if we go ahead and do the Punnett square, we will see these are the possible genetic makeup for their kids. Okay. So we're going to have 50% chance type A blood, 25% chance type AB, and 25% chance, 25% chance of type B blood. Okay. All right, let's try this next one. Okay. Now we're gonna actually do two things at the same time. We're gonna take a break as well. And when we come back, we'll take up this question. I'll see you in a little bit. We'll resume at 1:40. Okay. Hello again. This is the quiz one information. Just want to go through that with you. The quiz is going to be next Tuesday, which is May 24th, I believe. Yep, May 24th. It's going to be available from 6 to 11:59 p.m. Time will be 40 minutes. Just a reminder, once you start the quiz, the timer does not stop. Okay, so you have to finish it in one sitting. It will only be on the cardiovascular system. And just like last semester, you will click on this link on the day of the quiz, and it will take you directly to it. There will be 15 multiple choice questions, 4 application questions, and 4 multi-select questions. The multi-select questions are 2 marks each, and it will be worth 27 marks in total, which makes up 10% of your final grade. Okay. If you're registered with CALS and you need extra time for assessments, please send me the documents before Tuesday so I can accommodate you accordingly. If you send me the document last semester, you have to resend them, okay? Because this is a different course, and I will need to see the documents one more time. Okay. Any question about the quiz? Okay. So back to this question. Anyone have any answers that they want to share with the class? What's the possible blood type for question mark right here? Okay, let's find out. So to do something like this, we would write down the genotype of all the people we know for certain first. So type O, we know type O is little I. There's no other choice for type O. And for type A, we know there must be at least one IA and then something else. For type B, we know it's going to be IB something. For type B here is IB something again, and then IA something. Okay, so that's about as much as we can do without putting too much thoughts into it. Then we gotta try to use the intergeneration information to figure out, the missing alleles. So this lady here is type O, and so she has the little I and the little I. Where do the little I's come from? Well, one of the little I must have came from her father. Okay, so that's how I know that this father is IA little I and not IA, for example. The other little I must have came from the mother. So for the same reason, we now know the mother is IB little I. Over here, let's take a look at this. Okay, so there is an IA, which we know is not from her father. Her father could not possibly have an IA allele, right? Because otherwise that would change his blood type to AB. So this IA must have came from her mother. Okay. So where did this person, question mark, where did question mark get her IA? Well, there's only one source for it, right? Which is right here. So that means this other allele must be from one of these things. Okay. So are we able to definitively say whether she is IAIB or IA small I based on what we have here? And we know there must be a B or a small I here, but do we know for sure? Do we know for certain which one it is? Let's run a poll and see. If you think it must be IAIB, choose A. It must be IA small I, choose B. If you can think it's A or B and we don't know for certain, please choose C. If you're just not sure what's going on, you can choose D to let me know as well. Okay, kind of all over the place here for this one. Well, the answer is actually C. Okay, we do not know. We don't know if she is IAIB or IA small I, based on the information that is given to us. Right. All we know is that she passed on the IA to the next generation. She passed on the IA to her daughter. Right. So whether she's IAIB or IA small I, it doesn't change the fact that she passed the IA to her daughter. Okay, so in other words, what kind of allele is on this location is irrelevant based on the kind of information we are given. Okay, whether she's AB or just AI, we really don't know. Okay, so the question says list all possible blood type, which would be IAIB or IA small I. Okay. How about let's say they have another kid. Okay, let's say they have another kid. This time they have a son. And what if I say the son is type B now? Type B. Okay, if the son is type B, will we be able to tell now whether she's IAIB or IB small I? What do you think? Let's do another poll. The choices are the same. Okay, thank you for those of you who participated. We still won't know, okay? Because, okay, let's think about this. If this person is type B, right? Yeah, so clearly they inherited the IB from the father, okay? And what about the other allele? Well, this time, it's not going to be the IA that they got from the mother, right? They would either have to inherit an IB or a small I. Okay, whether they inherit an IB or small I from the mother, it would not change their blood type, right? In other words, we still cannot decide whether the mother is IA, IB, or IA small I, because both cases will result in a type B child. Okay, so even if this is the case, these are still the possible blood type. Okay, so, let's say they have another child, let's have another daughter, why not? And this time is type O. Now, will we be able to tell for certain whether she is IA, IB, or IA small I? And hopefully you will agree with me that if we have a type O child, then we can tell, right? Because we would need to have the little I, right, from the parent. Right? So if there is a type O child, then definitively she is going to be IA small I and not IA, IB. Okay. How will the test question look like? You are wondering. Well, here's one. Let's give that a try. Oh, I don't understand why they all changed to numbers. So weird. Okay, I'm just gonna wait a little longer. I only have 5 responses. All right. Let's see what's going on here. We're trying to find a genotype for John. John, where's John? John is right here. All right, so let's do the obvious ones. Add here is I, little I. Sue is little I. Mary is IAIB. Okay. Harriet is IA something. And then we got the Jane and the Tom. We'll worry about them later. So let's look at Harriet. Harriet has one IA, and the IA probably came from Mona, right? Not probably, definitively came from Mona, because the father Ed only has little I, right? And for that reason, just like the Josh question we did before, Harriet is going to be IA small I. Okay. Any question about what I've said so far? Okay. So what about John? Well, one of the small I in Sue's genotype came from Harriet. Okay. Which means the other small I must have came from John. Okay, one allele from each parent, right? Let's take a look at Mary. Mary has IA, so IA came from her mother, which means the IB, oops, wrong color, which means the IB must have came from John. So what is John's blood type? IB little I for sure. Okay, it's not going to get more difficult than this. All right, and at most you're going to have 1 or 2 questions like these on the test. So rest easy. Let's try one more just to be safe. What is the blood type of Mona? Okay, just a little longer. Okay, well, let's, I'm just gonna use the same, you know what, let's explain it one more time. All right, so for Harriet, we know she is IA small I, right? Ed is small I, because she's type O, he's type O. So based on this information, all we really know is that the IA must have came from Mona, right? So she is IA something. The question we have to try to figure out the answer is, you know, is this IA small I, IB, which one is it? Okay, and do we know for certain? Okay, the answer is we do not know for certain, because all we know is Mona pass on an IA. I have no idea what this other one is, right? What this other one is will not affect her ability to pass on the IA, right? So this other one could be, could be another IA, could be an IB, could be an IA small I, right? So let's see if any of these applies. IA, IB for sure. No, we don't know if it's for sure. IB little I for sure. In fact, for sure not. Okay, it's definitely not IB little I, because if she's IB little I, there's no way they can have a type A child, right? So not that. Either IB or IB little I. Nope. Either little I or IA IB. Definitely not little I, right? If both of them are little I, then her, Harriet is going to be little I as well. Remember the 4 things you have to remember? The 4th one is that if parents are both parents are Rh negative, then all the kids would have to be negative as well, right? So it's not that. The correct answer is need more information. Unfortunately, nobody chose that. But the important thing is, does it make sense after I explain it? And if not, please ask me a question so I can clarify it for you. Okay, thank you. All right, let's move. I know this is a lot to take in. This is a very involved, but make sure you review the notes and, you know, contact me if you need extra help. We can work something out. And the good news is this is not on the quiz next week, right? Okay, this is going to be on your test. Okay, question 11. I know I did 11 in the recordings already, but judging from the response in the polls, I think it's a good idea to go through it again. Okay, so here we're matching the babies, huh? Okay, there's some mix up in the hospitals, and then people don't know which baby is the one. So how do we figure it out? First of all, if there is a mix up at the hospital, we'd use DNA fingerprinting to figure out who's the parents, right? But just for the fun of it, we're going to use blood type to figure it out here. So here, we have A positive, B negative. Okay, so we're not going to start with that. Okay, to do these kind of questions. And by the way, matching question could show up on the test as well, right? So make sure you understand how to do these things. I'm going to start with parents who are O or AB, okay, because of the 4 rules that we talked about. So let's start with number 2 here. Is mixing up babies still an occurrence these days? I imagine so, but you know, I sometimes I read on the news that, you know, they find out they have been raising the wrong kid, right? Traumatizing. Yet I assume it still happens. O negative, right? So if both of them are O, then their baby also has to be type O. If both of them are negative, then their baby also has to be negative. So the answer is L. Okay, without question, definitely L. Okay, so 1 baby down, 3 more to go. AB negative and O negative. Okay, AB negative. If you are AB, you cannot have O babies. So that's gone. That's not yours. Don't touch that. If you're O, you cannot have AB baby. So that's not your baby. Okay? This one's already taken. So the only baby left from this selection is baby H. Okay? Try to pay attention to the logic I'm using, okay? So this one's gone, this one's gone. Now 2 more baby up for grabs. This one, O negative and B positive. If you're O, type O blood, you cannot have AB baby. That's not yours. So the only one left is I. And then that means J is theirs. Okay. I know it seems easy when the teacher does it, right? But the question is, are you able to follow the logic and repeat this on your own? All right. Let's find out for the next one. Okay, why don't you give this a try? Give you a little bit of time to try that. Okay, anybody want to share the results? You can list the alphabets in the order that you think it should be. And then we can talk about it. Don't list the blood types. List the alphabets. Like, is it G, F, E, H, H, F, G, E, whatever you think it is. Okay, write down. Okay, so we have H, G, F, E. Okay, H, G, F, E from Moira and Tyler. Anyone else have something different? All right, let's try to do this. All right, and again, the trick here is to find parents with A, B, and O first and start with those, because you can eliminate a bunch of answers right off the bat, right? So here we got a mother with A, B. Who will they not be taking home? If you're AB, you cannot take home the O baby. Okay. If you're type O, you cannot take home the AB baby. 2 babies left. Which one are you taking? If both of them are negative, their kids can only be negative. For that reason, the correct baby for this couple is H. Okay, H is gone. Next one, A positive and O positive. Okay, so let's see. If you're O positive, you cannot take this one. You cannot take the AB. Okay. Can O positive be your baby? Yeah, could be. I mean, if the mother is IA small I, it's possible this is your baby. Can A positive be your baby? Yeah, that's possible as well. So right now we don't know. We don't know. We're gonna put F or G. Okay, we do not know for certain yet. We know it's not the first one, but it could be the second or the third. Okay, O negative and B positive. If you're O, you do not take AB. I feel like I've been repeating that all class. And then if you are O and B, it's not possible to have a type A child, right? You need to have an IA somewhere, and neither of the parents have that. So that's not their baby. And so the only one left for them is F. And since F has to be couple number 3's baby, they cannot be couple number 2's baby. So it's G for the second couple, and the last one is E. So that's how you do it. Okay. Please figure it out. All right, how to do these questions, and again, contact me if you need extra help. Right? Sometimes you might even want to try to make up a question on your own to see if it works. Okay, feel free to email it to me if you made up a question. If it's a good one, I will maybe put it on the test or something. Okay, moving on. Do we have more polls? Let's see. Yes, we have more polls, but we also need to fill in this thing. So let's keep going for now. Okay, these are blood typing tests. If we're doing in class lab, that's something that we'll be doing as a simulation. And so over here we have a blood sample, and we drop it and mix it with antibody A, we mix it with antibody B, and then anti-Rh antibodies. And we want to see if a reaction occurred. So over here, we can see there is a reaction. All these clumpings are happening with antibody A. So we know this person is going to be A something. Okay, does it have a B? No reaction with B, so no B antigen. And there's a reaction with the Rh. So now we know this person is A positive. Okay, so what you're seeing here in this well is what we call agglutination, right? Agglutination, which is a clumping that occurs as a result of cross reaction. A cross reaction is what takes place when the antibody of the recipient is reacting with the donor's RBC, right? So this one is A positive. For the next one, no reaction with antibody A, so no A. Reaction with B, I'm gonna put a B. Reaction with Rh, we're gonna put a positive. All right. No reaction with A, no reaction with B. So that's going to be type O, because there's no A and there's no B. But there's a reaction with Rh, then it's O positive. For the last one here, there are reactions with both anti-A and anti-B. So we know the person is AB. No reaction with Rh. So that is AB negative. A little bit more here. It might be a little bit hard to see, but I promise you the ones that are shown on the online test will be clear. But you can see there are some clumping here. If I zoom in here to show you, it's a little bit of fuzzy stuff here. That's the clumping. So no clumping in A or Rh, but there is clumping in B. This is going to be B negative. Clumping in all three, AB positive, AB negative, and O positive. Okay. That is that. Okay, there is the fill in the blanks, which I will post the answer for you afterwards. You can check that yourself. Otherwise, we're not going to have enough time. Let me just put it here first before I forget. There we go. Okay, you can check that on your own. Perfect. Okay, long answer questions. Do we have any requests for long answer questions? Please let me know. Number 6. Sure. Number 7. Okay. Anything else? All right. Oh, and number 4. All right, let's go through and the application question for sure. Yeah. So let's do number 4 first. Is it safe to transfuse A negative RBC into a person with AB positive blood? Justify your answers. Okay. So let's do that. Copy the question. And we'll do it here. Okay. So we're taking A negative blood and then giving it to AB positive. Okay, save or not save. This is the donor, right? Donor, and this is the recipient. So we have 3 questions we have to ask, right? Number 1, what is on the donor's RBC? So we're going to draw the donor's RBC. Okay. A negative means there is A on the surface, and that's about it. There's no B, there's no RH. For the recipient, who is AB positive, what kind of antibody will the person have? Well, they're not going to have anti-A because they will end up attacking themselves. They will not have anti-B, and then they will not have anti-RH. So no antibodies for these blood groups, which means if you do the transfusion, there is no antibodies that will recognize the A on the surface. There is no cross reaction. Okay, no cross reaction. Therefore, safe. Does that make sense? Good. What was the next one? Number 6. Explain how RH sensitization could result in hemolytic disease of the newborn. Oh, I'm gonna go all the way down here, not enough space. Okay, so this was explained in tremendous detail in the lecture recordings. So make sure you watch the full version. But here I'm gonna give you the Coase Note version, okay, the condensed version. All right. So first of all, what is RH sensitization? RH sensitization occurs when a person with RH negative blood is exposed to RH positive blood. Okay. So that could happen due to wrong transfusion. It could happen during pregnancy. All right. So the hemolytic disease of the newborn is related to the pregnancy. So let's say this is the mother's bloodstream. Mother's bloodstream. And we are going to have the fetus, the baby's bloodstream. I never know if I should add another S here. Fetus's blood. In any case, the baby. And what separates the mother's blood from the fetus blood? Anybody know? What do we call that?
There's the placenta.
That is the placenta. Very good. Okay, so the placenta is pretty good at keeping the mother's blood and the baby's blood separate. Okay, so in our situation here, the mother is Rh negative and the baby is Rh positive. Okay, probably the father is positive. That's why we have this situation. So the mother's blood would look like this. We're just focusing on the Rh. I'm not looking at the AB. Okay, so no antigens on the surface. And for the baby's blood, there's going to be Rh on them. Okay, so Rh on them, Rh. Okay, so this is the first pregnancy. So what happens during the first pregnancy is that maybe the mother had some kind of trauma. Okay, maybe she fell. Okay, maybe there are other reasons that causes the placenta to tear a little bit. Okay, or it could happen during the actual delivery. You know, the placenta rupture a little bit, which is kind of common. And what happens is because of that, the fetal blood is now going to be able to enter the mother's bloodstream. Okay. So the mother who is Rh negative all of a sudden has been exposed to Rh positive blood. Okay. So now the mother has become sensitized. Okay. So once sensitized, she will start to make anti-Rh antibodies. Okay. I mentioned this in the lecture again in more detail, but briefly, for the ABO blood group, it's different. Okay, like if you're type A blood, you are pretty much born with anti-B antibodies. If you're type B blood, you're born with anti-A. But with Rh, you won't start to make the antibodies until after you've been exposed to Rh cells. Okay, just like you're not going to start making anti-chickenpox antibodies until after you've been exposed to chickenpox, right? So it works similar in that way. So once the mother is exposed to the positive blood due to the damaged placenta, either during pregnancy or during delivery, she is now sensitized and she will make antibodies against Rh. The whole process takes some time, okay? Making antibody is lengthy. It takes like maybe a week or sometimes week and a half or 2 weeks. So if this happens during delivery, by the time the mother has the antibodies, the baby would be out. And the baby would not be affected. Okay. However, let's say we have a second pregnancy now. So second pregnancy, same situation. Right? We have a mother with type negative blood. Same mother. Okay. Same father, assuming, and that's going to result in another baby who is also going to be Rh positive. Okay, and now we're gonna have a brand new placenta. Okay, and then we're gonna have the mother's blood. Then we're gonna have the baby's blood. And baby's blood has Rh positive, so there is all these Rh on it, right? So what's the difference between now and before? Well, because the mother was sensitized previously, her plasma is going to have these anti-Rh antibodies, right? Okay. So placenta is pretty good at keeping out red cells, but it's not very good at keeping out antibodies. So the antibodies will actually move through the placenta. Okay, and this actually is a good thing usually because let's say the mother has receive her COVID shot, right? And she has anti-COVID antibodies. We want those antibodies to go into the baby, right? So when the baby comes out, they're gonna have a little bit of immunity, right? We'll learn more about that next class. But in this case, these are antibodies that recognizes Rh. So once they cross the placenta, they will attach onto the baby's red cells and they will destroy them, resulting in hemolysis. Okay, the cross reaction, the clumping, the agglutination, which then results in hemolysis. So all these red cells are going to die. And that's going to lead to things like, edema. It's going to lead to anemia, right? Enlarged spleen and liver, because that's where all the damaged red cells are going to be recycled, right? And the entire set of symptoms, these group of disease, is collectively known as hemolytic disease of newborn. Okay, and it's not just Rh that could result in this. Okay, there are other blood group mismatch that could cause this problem. Okay, if you're interested, another one you can look up is Cal. Right, Cal mismatch can also be pretty severe. All right, so that's the answer for number 6. Okay, that's how sensitization could result in hemolytic disease of the newborn. So how do you prevent something like this from happening? That's the question number 7, right? Number 7, explain how RhoGAM prevents Rh sensitization. So in order to prevent this from happening, you got to do it in the first pregnancy. Okay, if you suspect the baby is going to be Rh positive, you have to suggest to the mother to take RhoGAM. And then how do you know if the fetus is going to be positive? Well, if the mother is negative and the father is positive, then there is a chance, right, that the baby is going to be positive. If the mother is negative and the father is negative, no need for RhoGAM. Okay. If the mother is positive, also no need for RhoGAM. Only negative people can become sensitized, right? Okay. So what happens is RhoGAM, pick a nice color here. Let's use green. RhoGAM, these are artificial anti-Rh antibodies. Okay, they're made in the lab and their job is to destroy anything that has Rh in them. So you're going to give it to the mother, okay, at like 28 weeks of pregnancy, and then again after delivery. You're going to give them these artificial antibodies that would find anything that has Rh on them and destroy them. Okay. So in the event that this takes place, in the event where the placenta is damaged during delivery, for example, and the baby cells go into the mother bloodstream, all these RhoGAM would destroy all these baby cells before her immune system is activated. If her immune system is not activated, then she is not going to be sensitized. And in the future, we're not going to have this problem because these antibodies were never made, right? So that's how RhoGAM prevent sensitization. Okay? And you might be thinking, wait, hold on a minute. Okay, if you give the mother a RhoGAM, wouldn't these antibodies cross the placenta just like here and end up attacking the baby's blood? That is a valid question. But remember, these are artificial anti-Rh. So the people who make them, obviously they're really smart and they know about this problem of crossing the placenta. So they actually make the antibodies really big. Right, so big that they will not be able to cross the placenta. They will only stay in the mother's bloodstream and therefore not affect the baby. Okay, so RhoGAM is useful for preventing sensitization. It is not useful for reversing sensitization. If the mother is already sensitized and she already made her own antibody, there's no use, giving her the RhoGAM. So is that okay for question 6 and 7? Whoever asked them, I forgot, sorry. Any follow-up questions about all these? It's very interesting stuff, actually. I think much more applicable than maybe last semester stuff. Semester 1 seems to be a little bit abstract, but hopefully this is more relatable. Any question at all? Okay. Application question. Bone marrow transplantation can be used to treat leukemia. Speculate the effect of bone marrow transplantation on a person's blood type. Justify your answer. So bone marrow contains the stem cells that are going to specialize into your blood cells, right? Through hematopoiesis. Okay, that's a term that we talked about in lecture. That forms all your red cells. So if you receive a bone marrow transplant from another person that has compatible that is compatible with you, then it's possible that your blood type will change. Let's say you are a type B person, and then you receive bone marrow transplant from a type O person. Okay, so all the new red cells that you make after receiving transplant could become type O. Okay, just because all the stem cells will be different now, right? Yeah. That actually happened to one of my students. She told me how she had a bone marrow transplant because of leukemia at a young age, and afterwards her blood type changed. So that's something that actually does happen. Number 2, Sally's first child was born with hemolytic disease of newborn. She learned about RhoGAM from her friend. Sally is now pregnant with her second child, and she is requesting to receive RhoGAM shot from her doctor. Is Sally's request valid? Should we give her the RhoGAM this time around? Tyler said no. Okay, what about the rest of you? Do you think we should do it? Should we give her the RhoGAM just in case there's going to be hemolytic disease newborn again? Okay, so we have a few resounding no. That's right, do not give her the RhoGAM. Okay, it's not going to help the situation. Remember, RhoGAM needs to destroy all the positive cells before her immune system is activated, right? Okay, but if her first child had hemolytic disease of a newborn, that means she already had the antibody herself. That means she was already sensitized, right? So giving her the RhoGAM now will not reverse that process. So my question to you is, how is it possible? How on earth did she get sensitized before her first child? Right? How is it possible for someone to already have the antibody in her system before she even was pregnant? Anybody can answer that? Do you understand my question? In order for her first child to have hemolytic disease of the newborn, Sally must have had the anti-Rh antibody in her system already, right? So how did she get those antibodies in the first place? And Tyler said bad blood transfusion. That's correct. Okay, maybe she received positive blood transfusion sometime in her life. Okay, and that does happen sometimes. Okay, like if the person is like bleeding out, okay, and then we don't have any negative blood available, and we know for a fact that the person did not have a positive transfusion in their medical history, then they will give them positive blood, because if you don't give them the positive blood, they're gonna bleed out and die, right? And it's safe to do it for once, okay? But once that happens, then she's gonna become sensitized and she's gonna have the antibodies, right? And that's why during the first pregnancy, those antibodies could now cross the placenta and affect the baby. Okay, how often does that happen? Oh, in terms of like wrong transfusion or deliberate positive transfusion, it happens more often than you would think. Because remember, right, it's not just Rh that we have to worry about, right? There are many blood groups that we haven't touched on, right? And sometimes it's just not possible to find those match. You know, in an ideal situation, if someone's gonna have surgery, you know, like next week or something, the hospital is going to make sure you're going to have those type of blood available for those people in case they need it, right? But sometimes if it's like a really rare one, and then, you know, maybe something went wrong in the surgery and the person is having excessive bleeding and you run out of that rare blood type, then maybe you would have to give them the positive. Okay. And again, when I say positive, I don't just mean Rh, right? Like I talked about Cal earlier. There is Cal positive and Cal negative. Same situation. If the person is Cal negative and you give them Cal positive blood, you will sensitize them for that particular blood group. Okay, so that's why sometimes like right before transfusion, they have to do a complete blood test on you to make sure that you were not previously sensitized for some of these groups, right? Especially if your medical record is not complete, right? They just have to be sure, right? Because otherwise, if they thought you were not sensitized and then they give you like a wrong blood transfusion, that could cause quite a bit of problems. Okay. So I know it's very demanding, this lecture, a lot of moving parts and a lot of things to think about and memorize, but I'm here to help. So again, if you need extra help, please feel free to reach out. We can do it over Zoom, we can do it over email, whatever works for you. But the most important thing for you now is to study for the quiz next week. And like I said, the first quiz, I try to make it more friendly, if you would. Hopefully it's not gonna be too difficult. Now, before you go, if you want to go, not a problem at all. But let's just play like a Kahoot game for review for the first quiz. Okay, I forgot, and there I lost a person immediately. It's like, oh, Kahoot, let's run away. Anyways, let me let me try to find the thing here. I forgot to log on before I come into class. Give me a second, okay? How's the class going so far anyways? Like, do you guys find this to be more difficult than last semester, or just trying to have small talks with you guys while I look for the Kahoot? Where is it? Oh, I gotta click through all these things. Okay, there we go. There we go. Yeah, certainly more things to explain, right? In this one than the previous one. Yeah. Okay, let's go classic mode, and I have to share this screen with you. Screen 1. There we go. Okay, so please go to kahoot.it, type in the game pin. You can also scan this QR code. Okay, and then we're gonna do this. Okay, so you just type this into your browser. Copy that into the chat box for anyone who wants it. You can also do it on your phone. You just have to scan the QR code. Okay, I'm just gonna wait a little bit more, maybe like a minute or so, in case people are still trying to join. Is anybody trying to join but not able to do so? Or is this it? I'm gonna start then. All right, here we go. I think you can still join once we start, but we're gonna get going. Yeah, the human circulatory system is closed because the blood stay inside the vessel at all times, and it's two circuit. We have the systemic circuit, and we have the pulmonary circuit. Okay, very good. Next question. Where's the next button? No, my video is blocking it. There we go. Next one. The heart wall has 3 layers. The endocardium is the innermost layer that is in direct contact with the blood. The myocardium is the muscle itself. That's the thickest layer of the heart wall. And then we have the epicardium, which is on the outermost layer, and that is continuous with the sac that goes around it, the pericardium. Okay, so the innermost is the endocardium. Oxygen-rich blood. So you can refer to the diagram we drew last class, right? This one, right? Let's shows you what's oxygen-rich and what's oxygen-poor. And over here, the aorta is the one that is rich in oxygen. Okay, a lot of people also choose pulmonary artery. Remember, the pulmonary artery takes the blood to the lungs for reoxygenation, right? The whole purpose of taking it to the lungs is so that you can add more oxygen to it, right? So pulmonary artery actually is oxygen poor. Next one. Yeah, almost everybody got it. Very good. The hepatic takes it to the liver and the pulmonary takes it to the lungs. And renal takes it to the kidneys. Blood flow is the slowest in the capillaries because once again, you have to slow down substantially in order for the gas exchange to occur, right? The capillary exchange, all that stuff needs to happen, you know, when the blood is slowed down substantially. If it's just zooming by really fast, you won't have time to do it, right? I guess the people who pick veins, you might be thinking, the blood pressure. The blood pressure is the lowest in the veins, but this is asking for the blood flow, and the speed is the slowest in the capillaries. Think about the 3 graphs that we drew last time, right? Where is it? Right here. Okay. And again, the best way to study for this is just to print out a blank study guide and then just do it yourself. Okay. And then you know, look at some of the poll questions that we did last class. That's probably the best way to do this. All right. Yep, the tricuspid valve is the one that separates it on the right side, 3 flaps, and then the bicuspid or the mitral valve is the one that separates them on the left side. Okay. Yeah, the SA node is the natural pacemaker. Very good. Stroke volume. Let me just refresh your memory with the formula here. Cardiac output is stroke volume multiplied by heart rate, right? And you can see all the units here. So for stroke volume is how much blood you squeeze out per beat. So that is milliliters per beat. Okay. Yeah, milliliter is volume, right? So volume per beat. I always wonder how they calculate those marks, those scores. This is right from the PowerPoint slides, and it shows atherosclerosis. We have the atherosclerotic plaque here, and then we have the blood clot that is forming over that. Okay, so that's the answer. It's not heart block, right? Heart block we talk about, it's an interruption in the signal from SA node to AV node and so on and so forth. Congestive heart failure is when your heart fails to empty the chambers. And pericarditis is inflammation of the pericardium. Correct answer is atherosclerosis. Okay, just a couple more. So at P, it is the atrial contraction. And this is when the signal is going from SA node to AV node. And at R is the ventricular contraction. Okay, so it's asking for P, which means this is the answer. Angioplasty is to reopen, partially block blood vessel, where we talked about that last week. Removing damaged blood vessel. We actually did not talk about that at all. So that wouldn't be the answer. Yeah, angioplasty. Okay. Two more. Good. When you exercise, your heart rate goes up. And since cardiac output is dependent on heart rate, when heart rate goes up, cardiac output also goes up. All right, here's the last question. We actually did this last week, okay? It's one of the application questions that we took up, and the correct answer is pulmonary edema. For the explanation, please watch the recording from last week, okay? So hopefully this gives you an idea of where you are at in terms of how much studying you have to do. Yeah, thank you for participating, and well done, all of you. And let's find out who won. And we have Mitch, Moira, and Tyler at the top here, I think. Very good. Okay, so that's it for today. Okay, thanks again for coming. I will see you all next week. Right, have a fantastic weekend. Bye now.